research involving recombinant and synthetic nucleic acid molecules with our resource speaker, Dr. Cesar Nadala, the other half of Dr. Lourdes Nadala. I want to welcome all of you here today and thank each of you for participating in this activity. This webinar series will run through next year, and we are fortunate to have 7 Balik scientists who will share their expertise in virology. So the DOST VIP program was conceptualized to address the pandemic readiness of the country. The ongoing COVID-19 pandemic presents many concerns, particularly effective clinical and public health management, primarily on novel viruses. And these issues can only be addressed using science and technology through research and development. So the objectives of the VIP program or the Virology and the Vaccine Institute of the Philippines, it will serve as the premier research and development institute in the field of virology, encompassing all areas in viruses and viral diseases in humans, plants, and animals. And second, it will act as a venue for scientists, both here and abroad, to work collaboratively to study viruses of agricultural, industrial, clinical, and environmental importance. And third, establish strategic partnerships with the world's leading scientists, virology centers, and institutes, and conduct innovative and pioneering researches that will advance the frontiers of virology in the country. So to institutionalize the VIP, we get support from our lawmakers both in Congress and Senate, by which the bill would be for enactment for the establishment of the VIP. So the bill was already approved at the lower house in the second hearing, and we are still waiting for the Senate to convene. And we are still, while waiting for the VIP bill to be enacted into law, the DOST has initiated several R&D projects which is being implemented by ITDI. So these projects are being implemented in partnerships with several local and international researchers and institutions. With these initial projects, we hope to build the capacity of the VIP and help resolve some of the pressing issues in the country brought about by these viruses. And part of the project's activities is engaging the expertise of the Balik scientists to help us accomplish these initiatives. Dr. Lourdes Nadala will discuss this morning the proper handling of multi-drug resistant bacteria. I thank Dr. Nadala for accepting the DSP engagement and helping ITDI in the VIP program. My special thanks also to the VIP team of ITDI, the Technological Services Division, and the Planning and Management Information System Division for organizing this event, the Balik Scientist Program Secretariat of the Philippine Council for Health, Research and Development, and also to the Philippine Council for Agriculture and Aquatic Research and Development, to our ever-supportive Secretary Fortunato T. de la Peña, Undersecretary Rowena Cristina Guevara, Director Montoya of PCHRD, and Director Ibora of PICAR. I encourage all the participants to join in the question and answer portion actively, and I hope you will learn a lot from this webinar. Again, welcome and a pleasant morning to all of you. Thank you.
Thank you very much, Dr. Briones, for that warm welcome to our speaker and guests alike. But before we proceed to the meat of our session this morning, let me first introduce our distinguished speaker for this morning. Our speaker obtained her bachelor's in biology and a master's in microbiology at the University of the Philippines, Los Baños. She then pursued a UNESCO postgraduate course in microbiology at the University of Tokyo, then a doctorate in microbiology at the University of Hawaii. She is a microbiologist and virologist by training. She has over 15 years of experience in microbial systematics and culture collection related activities. During her doctorate studies at the University of Hawaii, she developed the first continuous cell line from shrimp. Upon her return to UPLB in January 1997, she collaborated with the Bureau of Fisheries and Aquatic Resources to help alleviate the infectious disease problems in aquaculture. In year 2000, she then pursued her postdoctoral research at the University of Cambridge on a viral discovery project and chlamydia diagnostics, where she also managed a BSL-3 facility. In 2002, their laboratory at the University of Cambridge established a spin-out company in the Bay Area in San Francisco, California, USA, called the Diagnostics for Real World Limited. She has co-authored numerous articles and published in referee journals. She was also a reviewer for the Journal of Virological Methods in Aquaculture and an external reviewer for a PhD dissertation at the James Cook University in Australia. It is my pleasure to introduce a pride of our country, our very own DOST Balik scientist, Dr. Lourdes M. Nadala, to talk about proper handling of multi-drug resistant bacteria. A virtual round of applause, please. Good morning, Dr. Lou.
Good morning, everyone. And thank you so much, Meg, for that very good introduction. And I also would like to thank Dr. Briones and the ITDI family, who has been very supportive and very helpful in all our questions and all our requests. And it is an honor and a privilege for me to be part of this Balik Scientist program, to be able to share whatever I have learned in my over 40 years of work as a microbiologist and as a virologist. So should I go ahead, Meg?
Yes, ma'am. Go ahead po. And if you can share your screen po. Thank
Okay.
you.
Select mo yan. Tapos, share mo dito. Hindi yun yung i-select mo. Tapos, share. Okay, tapos i-run mo na yung PowerPoint.
Okay. So this morning I'll be talking about multi-drug resistant bacteria, and you might wonder why it's being part of the Virology and Vaccine Institute of the Philippines. I think it is because everybody realizes that MDR is becoming a global threat, and before it becomes into a massive problem, everybody and all countries are already having their action plans to be able to handle multi-drug resistant bacteria. So when I was asked to give a seminar on this topic, I had to go back to my microbiology hat, because my experience with MDRs before was when I was handling them as accessions in the culture collection at UPLB. It has come a long way, and I think there are now a lot of studies that have been conducted on multi-drug resistant bacteria. So I would like to give a brief outline of my presentation, which will be for 2 days. I'll give a little bit of background on antibiotics and MDRs, and also talk a little bit about classes and sources of antibiotics, or antimicrobials as they are now called. And also it would be important to understand the mechanisms of action of these antibiotics to be able to understand how the bacteria is able to develop resistance against these antimicrobials. And then I would also like to go over the development of antibiotic or multi-drug resistance, because different organisms are able to develop resistance differently from each other. So I would go over the different mechanisms of development of multi-drug resistance by different types of organisms. And it is also important to be able to detect or differentiate the multi-drug resistant strains from the wild type. So I would go over some methods of detection that are being used in current detection of MDRs. And lastly, I would talk about the initiatives to control or to properly handle antibiotic or multi-drug resistance. Just to give you a brief background, there was a B debate meeting held in 2015 in Barcelona, Spain, and this was attended by various representatives from various countries, like the US Centers for Disease Control and Prevention, or CDC, the European Center for Disease Prevention and Control, or ECDC, and the World Health Organization, or WHO, among others. And at that meeting, everybody had a consensus that infections caused by multi-drug resistant bacteria is an emergent global disease and a major health problem. So in that meeting, everybody was in agreement that multi-drug resistant bacteria could be a major health problem because it's being experienced by low middle income countries and even developed countries. So everybody was already talking about action plans to be able to handle or control the further development of MDR. The focus of the B debate was on 3 things. They were discussing the antimicrobial resistance in animals and the food chain. And secondly, the antimicrobial resistance in the environment and the community. And thirdly, antimicrobial resistance within the healthcare setting. So these are the 3 major sources of or settings where antimicrobial resistance is happening. In terms of statistics, in 2019, 700,000 people died of drug-resistant infections annually, and these are just some of the countries that were cited. India, for example, had 58,000 deaths, and US, which is already a very developed country, had 35,000 people who died from drug-resistant infections. And the European Union has had 25,000 deaths associated with drug-resistant infections. And they are projecting by 2050 that if there are no interventions or drastic means, global death could reach as high as 10 million. Of course, everybody's trying now to address this multi-drug resistant bacteria. So hopefully this number is not going to take place, or we would be able to thwart any or decrease at least this projected number of deaths from drug-resistant infections. So this is just, I just wanted to show you this pyramid of infectious diseases. And the patient is in this left-hand side, which is the host. And then the drug is on the apex or the top. And then the pathogens are here. And you have also commensals, which are affected by therapy. So therapy here is the antimicrobial. So antibiotics could be a broad spectrum. So that means it could affect even the non-target organisms or like commensals or even the good bacteria, for example. So they have antibiotics have been very useful in treating diseases, and this one everybody agrees, and everybody has found the very important contribution of antibiotics in treating infectious diseases. But at some point, the target pathogens have developed resistance, and also the commensals or the non-target organisms, likewise, which have been affected by the antibiotics, have also developed resistance. So it is a chain reaction for the target bacteria and the non-target bacteria to develop resistance. And this is a natural phenomenon for for organisms, for that matter. So just like what we have now, like SARS-CoV, the original wild type has now mutated into several forms in order to evade the immune system of the target host. So likewise, MDR or multi-drug resistant bacteria had to evolve in order to survive in these settings. And the host also can develop resistance from pathogens, or from pathogens, and also from developed toxicity or experience toxicity from therapy. And so some hosts could have allergic reactions, for example, or side effects from the therapy or antimicrobials that they are taking. So there are several classes of antibiotics, and each of these class has a different way of affecting the target organisms or different mechanisms of action. So I'm sorry for this very small slide, but we have the major form of antibiotics known as beta-lactams. Beta-lactams were discovered, or penicillin, for example, was discovered in 1928 or around the 1930s. It's produced by penicillium, and it has been used extensively around 1940s. And there have been other types of beta-lactams, such as cephalosporins or amoxicillin. And these are all produced by fungi. And this type or this class of antibiotics inhibit cell wall biosynthesis in bacteria. So they are very effective against gram-positive bacteria because they interfere with cell wall synthesis. And then around 1940s, another class of antibiotics known as aminoglycosides were discovered. And among the famous members of this family or class are streptomycin and kanamycin. And the mode of action of this class of antibiotics is that it inhibits the synthesis of proteins by bacteria. So this could lead to death, cell death. And so these two classes of antimicrobials or antibiotics are bactericidal, meaning they really kill bacterial cells or they lead to cell death. And then around 1950, we had this new antibiotic then, chloramphenicol. And just like aminoglycosides, they interfere with the synthesis of proteins. And this used to be a first-line drug in developed countries, but because of the development of resistance by the target organisms, it has also been it's not extensively used as before. And then around 1960, there were glycopeptides, which is exemplified by vancomycin, and this also inhibits cell wall synthesis. So just like beta-lactams and aminoglycosides, this is also a bactericidal agent. So it can lead to cell death. And around around the same time, quinolones were also discovered, and examples are the ciprofloxacin, trovafloxacin, and this class of antimicrobials interfere with DNA replication and transcription. So this class of antimicrobials is also a bactericidal, consists of bactericidal agents. And then we have a bacteriostatic class of antimicrobials known as oxazolidinones, and it has the same mode of action as chloramphenicol. So they also interfere with protein synthesis. The next slide shows another set of classes of antimicrobials, and we have the sulfonamides, tetracyclines, and macrolides, which are all bacteriostatic. And sulfonamides, they do not kill bacteria but prevent their growth and multiplication, and they can cause allergic reactions to in some patients. And tetracycline, doxycycline are also considered as bacteriostatic. They inhibit synthesis of proteins by bacteria, preventing growth. Macrolides, such as erythromycin, inhibit protein synthesis by bacteria, but they are mostly bacteriostatic, but they can also lead to cell death. And we have these other classes of antimicrobials which have been discovered in 1960 up to 1980. So if you notice, the discovery of new antibiotics is lagging behind. So the last class was around 1980. So there is a need to discover more antimicrobials if we would really like to thwart or control multi-drug resistant bacteria. So just to give you a summary of their action. So for cell wall synthesis, these are the antimicrobials that are that interfere with cell wall synthesis, the beta-lactams, and then you have also vancomycin and bacitracin. And for those that interfere with nucleic acid synthesis, you have these sulfonamides and trimethoprim, which interfere with nucleic acid replication by interfering with DNA gyrase and RNA polymerase. And there are also a number of antimicrobials which affect the 50S subunit of the 70S ribosomal RNA, which plays a role in translation. And these classes of enzymes are the macrolides and chloramphenicol and streptogramins. And there are also antimicrobials which affect the 30S subunit of the 70S ribosomal RNA. So these classes of enzymes are have different modes of action. So they are used for specific types of bacteria. So if your target organism is gram-positive bacteria, most likely you would use antimicrobials which are interfering with cell wall synthesis. If you wanted a broad spectrum antibiotic, then you can use those that affect protein synthesis or nucleic acid synthesis. So I just wanted to emphasize the mode of action of these classes of antimicrobials, because later on, this will also be the target of the bacteria itself to be able to develop resistance to this specific bacteria, to this specific antimicrobial or antibiotic. So please keep in mind that, for example, antibiotics which are targeting cell walls are the beta-lactam antibiotics, which are the penicillins, cephalosporins, and carbapenems. So beta-lactam antibiotics are used for gram-positive bacteria. And then for glycopeptides, they also have the same target. They interfere with cell wall synthesis. So bear in mind those mode of action, because we will come back to them when we discuss the multi-drug resistant bacteria. This is just a summary slide for those that inhibit protein synthesis, which includes the aminoglycosides, tetracyclines, chloramphenicol, macrolides, lincosamides and streptogramins, and oxazolidinones. So some of the names are very tongue twisting. So then again, quinolones, which are inhibitors of DNA replication. And lastly, folic acid metabolism inhibitors. Folic acid is required by bacteria in their growth. So those that are affected, those antimicrobials which affect folic acid metabolism are the sulfonamides and trimethoprim. And this slide just explains that sulfonamides inhibit it inhibits a certain enzyme in a competitive manner for the enzyme than the natural substrate, which is the para-aminobenzoic acid. And trimethoprim acts at a later stage of folic acid synthesis. So these two antibiotics act in synergism to inhibit the growth of bacteria. So how do bacteria develop antimicrobial drug resistance? As I have said earlier, when I was talking about the mode of action of the antibiotics, those are also the same enzyme. Those are the same actions that are being targeted by the bacteria to be able to develop resistance. So, for example, the next few slides will just show you the natural causes of drug resistance. So we have what we call a selective pressure. So no matter what, all living organisms have the ability to mutate. So there is a natural cause for or a natural cause of drug resistance, for example. So in the presence of an antimicrobial or an antibiotic, microbes are either killed or which is shown in the upper part of this slide. So the non-resistant bacteria which multiply will die in the presence of the antimicrobial. However, drug-resistant bacteria are able to live or has the ability to develop resistance, which I will explain later how, and then multiply and then continue to spread. So the person remains sick because the bacteria is able to evade the effect of the antimicrobial or antibiotic. And then there is also a mutation that takes place in nature. So non-resistant bacteria exist in nature, but they also tend to mutate, as I have mentioned earlier. And then a few of these that mutate are able to grow in the presence of the drug or the antimicrobial. And then drug-resistant bacteria will multiply and thrive while the wild-type bacteria will die off. So this is one way of trying to develop multi-drug resistance. And a third way is through gene transfer or transfer of mobile elements. So from one bacterium, for example, who has developed resistance, this resistant gene can be transferred to a wild-type bacterium. And in doing so, the wild-type bacterium will get the antibiotic resistance marker and then multiply and then lives and continues to thrive, while the wild-type bacterium will die in the presence of the drug because it did not receive the antibiotic resistance marker from a donor cell. So this slide shows you how the gene is transferred from one bacterium or from one cell to another. And there are three ways, at least three ways. And one is through plasmid transfer. So if you recall from your genetics class, plasmids are extra chromosomal genetic material which are self-replicating. And this can be, most of the antibiotic resistance markers are found or are plasmid-borne or are found in plasmids. And this can be transferred from one cell to a non-resistant cell. So by transferring the plasmid, which contains the antibiotic resistance marker, this wild type is conferred antibiotic resistance. So it will also become resistant. And then another method is through phage.
Still working.
By phage delivery. So the phage carries the resistance marker and transfers it into a wild type bacterium, and then releases the genetic material which contains the the drug resistance marker, and that therefore this cell will acquire the resistance antibiotic resistance marker or gene. And then another method is through transfer of free DNA from another cell, and this is just a simple transfer of a genetic part which contains the resistance gene. So those methods I have just discussed were mostly a genetic way of acquiring resistance or antibiotic resistance marker. There are also other ways by which the bacteria are able to develop resistance or multi-drug resistance. And these are biochemical resistance mechanisms, which includes antibiotic inactivation or target modification. And some are able to alter their permeability, or so they have this structure in their cell wall known as sporins or channels. So bacteria are also able to alter the permeability features so that they are able to stop the entry of these antibiotics into their system. And they're also able to bypass certain metabolic pathways to be able to survive the effect of antibiotic or the effect of an antimicrobial. So antibiotic inactivation is shown by certain bacteria which are able to produce beta-lactamases. So if you recall in my earlier discussion, the beta-lactam group of antimicrobials, which include the penicillin and cephalosporin, for example, attack or interfere with cell wall synthesis, right? So certain enzymes are able to hydrolyze the beta lactams. So because of their they are able to produce beta lactamases. And there are several beta lactamases that have been discovered, and they have been classified either by structure or function. So according to the Ambler classification, there are 4 classes of beta lactamases. And class A, which is produced by the family Enterobacteriaceae, includes penicillinase. So if you have heard about the ESBL or extended spectrum beta lactamases, they are able to change substrate profile because of amino acid substitution. So they allow hydrolysis of most cephalosporins. So these ESBL are resistant to penicillins and the 3rd generation cephalosporins. So Enterobacteriaceae is composed of a lot of genera which are showing now multi-drug resistance. So they're becoming a very important family in terms of developing multi-drug resistance. Then you have the class B, which are called metallo-beta lactamases, and this requires zinc or heavy metals for their activity. So they are able to resist inactivation by carbapenems and aztreonam antimicrobials. So bacteria are able to also evolve so that they are able to survive in the presence of these drugs. So they're able to produce these enzymes which would inactivate or degrade the antibiotics. Then you have the class C, which are called cephalosporinases. This is produced by gram-negative bacteria except Salmonella and Klebsiella. So most of the gram-negative bacteria now are able to resist or are able to survive in the presence of cephalosporin because of this enzyme, which inactivates the antimicrobial or antibiotic. And then the last class is the oxacillin hydrolyzing enzymes. Again, these are found mostly in Enterobacteriaceae and in Pseudomonas aeruginosa. So these enzymes are able to inactivate beta lactamases, thereby allowing them to grow in the presence of this antimicrobial. The next important class of, or the next important mechanism of multi-drug resistance is shown by certain bacteria who are able to produce aminoglycoside modifying enzymes, or they're called AMEs. And these AMEs reduce the affinity of a modified molecule. So they modify certain enzymes, and then which interfere with the binding of the 30S ribosomal subunit. And so if once this enzyme interferes with the 30S ribosomal subunit, then translation and downstream protein processing is not going to happen. So these enzymes would inactivate this aminoglycoside type of antimicrobials or antibiotics. So these AMEs are identified or have been identified in Staphylococcus aureus, Enterococcus faecalis, and Streptococcus pneumoniae strains. So these are all gram-positive bacteria which are able to produce aminoglycoside modifying enzymes. And another enzyme that is produced by bacteria to allow them to resist or to survive antimicrobials is this chloramphenicol acetyltransferase. And this is found in a few gram-positive and gram-negative bacteria and in some of Haemophilus influenza strains. And again, these are enzymes which would interfere with or inactivate the antibiotic or antimicrobial, so it's not able to bind to the 50S subunit properly. So therefore, it cannot proceed with its action. So allowing the bacteria to grow or survive in the presence of chloramphenicol. Another mechanism is what they call as target modification. So what happens is there is an alteration in the 30S subunit or 50S subunit of the 70S RNA. So bacteria is able to have some modification so that the antibiotic cannot bind to the 30S or 50S subunit. Therefore, bacteria can still do protein synthesis even in the presence of this antibiotic. Another way is alteration in penicillin binding protein, which is found in gram-positive bacteria. So the penicillin binding protein is where the beta-lactamase or beta-lactam antibiotic would bind. But because of alteration, the antibiotic cannot bind properly to PBP. And so therefore, the action of the antibiotic is impeded. And this is found in Enterococcus faecium and Streptococcus pneumoniae, which are all gram-positive bacteria. And also in multi-drug resistant Staphylococcus aureus, this is the mechanism or the way that Staphylococcus aureus is able to resist or able to grow in the presence or survive in the presence of the antimicrobials or the multi-drug resistant Staphylococcus aureus is able to grow in several, even in the presence of several types of antibiotics. There are also ways for the bacteria to alter a cell wall precursor. So again, these are the gram-positive bacteria which are able to grow in the presence of vancomycin and teicoplanin because they're able to do to alter cell wall precursor. So the antibiotics cannot interfere with cell wall synthesis. And then I think this is the last target modification example where mutated DNA gyrase and topoisomerase leads to fluoroquinone resistance. If you recall, fluoroquinolones interfere or act on the cell through interference with DNA replication. So to be able to counteract or counter the effect of antibiotics, the organism itself has the ability to have a mutation in its DNA gyrase and topoisomerase such that these are not affected in the presence of the fluoroquinolone or the antimicrobial or antibiotic. And there's a last mechanism, which is ribosomal protection mechanisms imparting resistance to tetracyclines. Similarly, the RNA polymerase are also, or mutated RNA polymerase have also been found in some bacteria which confer resistance to rifampicin. So these examples or these properties give the bacteria the ability to counteract the effect of antibiotics or antimicrobials. And then another way, aside from biochemical means or biochemical changes, is through altered permeability. You have to keep in mind that antibiotics or antimicrobials have to go through the cell wall into the cell before, depending on what its action is. So for antibiotics which would attack or interfere with cell wall synthesis, they will interfere in the cross-linking of the peptidoglycan, for example, on the cell wall. So the cells will not be able to produce live, complete, or live cells with a complete structure. So other antibiotics have to go through the cell wall, through the outer membrane, into the periplasmic space, into the inner part of the cell, through channels, basically. So some of the bacteria are able to alter the permeability to these antibiotics, such as found in Pseudomonas aeruginosa. So they are able to prevent, for example, entry of these antibiotics into the cell. And also there are what they call as efflux pumps, which would be very specific to antibiotics and are mostly multi-drug transporters. So they're very specific to the transport of drugs into and out of the cell. And this can be altered, for example, so that the organism is able to counteract the effect of antibiotics. And this is one mechanism where the organism can acquire multi-drug resistance. And then lastly, they have what we call as bypass of metabolic pathways. So bacteria are able to also produce an alternative target. So it's like trying to have a how do you call that? It's something to allow them to bypass a part of a biosynthetic pathway, for example, so that they're able to still function in the presence of an antibiotic. So An alternative target allows the bacteria to survive by adapting the role of a native protein, for example. So it sort of mimics a native protein to allow it to evade the effect of the antimicrobial or antibiotic. And this is just another example of a bypass of a metabolic pathway. So all these examples or mechanisms show us that bacteria are able to develop resistance to drugs or antimicrobials with their own natural mechanism, or they sort of adapt because of it's like survival. So for example, most viruses are also able to do that. So they develop or they mutate to evade, for example, the flu vaccine. So the flu vaccine has to be changed every year because the flu strains are mutating every year, and they are very they are, if you recall your virology, they are segmented. So they have 8 segments in their nucleic acid. So these 8 segments can produce different combinations through reassortment. So then they have to develop a vaccine based on the latest strain, for example, of flu that went around the world, for example. So they come up with a combination of various strains of flu for next year's flu vaccine. Likewise, bacteria is able or are able to evade the effect of antimicrobials through several ways or means. So that makes them very adaptive to drugs or antimicrobials. And so this is the reason why it's worrying a lot of our public health organizations and around the world, because it's also affecting all countries across, regardless of whether it's developed or developing or low income or middle income countries. So it cuts across all societies. That's why it's alarming, basically, because it is affecting everybody. And so aside from all those natural or natural mechanisms of bacteria to be able to adapt to or to develop drug resistance, there are also other contributing factors such as societal pressure, for example, and then and this is just because of the use of antimicrobials, even when used appropriately, for example, creates selective pressure for resistant organisms. And then, of course, the most the culprit of them all is the inappropriate use of antibiotics. And in the past, I know this for a fact, that I could buy an antibiotic over the counter in the Philippines without a prescription. But I was happy to hear that I think a few years ago, there was a law enacted by Congress to require prescription for drugs to be, or antimicrobials to be able for individuals to be able to procure antibiotics over not just over the counter. And then of course, inadequate diagnostics. So if there is a misdiagnosis, then the antibiotics that may be dispensed will not be may not be appropriate for that organism. And then hospital use, as I have said, it has been used heavily in hospitals, especially in critically ill patients. So this can also worsen the problem by selecting for antimicrobial resistant microorganisms. And lastly, it's agricultural use. Antibiotics are also used in agriculture, especially in the animal industry, because they are used to promote growth. And so now they're trying to limit the use of antibiotics in the animal industry, for example, like in poultry and even in aquaculture. I think in the Philippines, which I will talk about tomorrow, they have also tried to regulate the use of antibiotics in animal husbandry and in aquaculture. And I think I don't have much time now, but I will be talking about the WHO priority list of pathogens for R&D of new antibiotics tomorrow. So if you have any questions, please let me know.
Okay, thank you very much, Dr. Nadala, for that in-depth presentation on the proper use of multi-resistant bacteria, multi-drug resistant pathogens. Okay, so let us now proceed to the Q&A portion. I'm sure that our participants are excited to ask some questions to our esteemed speaker. So we now have a first question from a student here via Zoom. But before we proceed to the Q&A portion, just a reminder for everyone asking about the certificate and the copy of Dr. Nadala's presentation. We will provide it tomorrow since we still have a part 2. And please stay tuned for the part 2 so that you can answer the evaluation form so that you can secure a copy of your certificate and the presentation of Dr. Nadala. So let us proceed with the first question. This is asked by Neil Atienza. Good morning, Neil. Kaapelido ko pa. So his question is, hi, I wanted to ask if there are developments in the literature to prevent such microorganisms in developing these enzymes, perhaps through genetic engineering in order to develop microorganisms for laboratory use.
I don't quite understand what he meant, what he means, he or she means by this. So if the student is asking, what's the name of the participant? Neil?
Yes.
So if Neil is asking if you're able to develop a microorganism which is not able to produce these enzymes, for laboratory use, of course, you can. But for in the real world, these microorganisms are brought about by infection. So they can be naturally occurring already. So, but if you want it for lab use, of course, you can develop those by genetic engineering for lab use.
Okay, so that was a question from Neil. He has a follow-up here. Of course, the characterization of the molecular organisms for the development of drug resistance are first required. Are information regarding these available?
Yes, I will talk about them tomorrow. So it is very important to diagnose correctly. So we will be talking about detection methods tomorrow, and also what should we do about in terms of research to be able to use an alternate way of treating multi-drug resistant organisms. So we should not be relying only on antibiotics, because development of antibiotics from concept to market could take like 10 years, for example, because of the regulatory hurdles that the manufacturer has to go through. So there are alternate ways, which I will talk about tomorrow morning.
Okay, thank you, ma'am. So we have another question from MMSU. This is asked by Harold via Zoom. His question is, can a resistant bacteria be killed with soap and bleach? Soap with water and bleach.
Yes, I would think so because, for example, gram-negative bacteria are very susceptible to bleach and detergent because this detergent would hydrolyze the cell wall or the cell membrane of bacteria. So I think the practice of washing for 20 seconds is very effective against viruses, against bacteria, and it's really a good practice to maintain, which we have done so very well in this pandemic.
Okay. Thank you for that question, Harold. We have another one from Rafaela Lau via Zoom. Her question is, hi, Doc Lu Nadala. Thank you for that wonderful presentation. Do you think we should consider incorporating the screening of pathogenic bacterial resistance genes in Philippine hospitals as part of offered services to help mitigate the growing number of MDRB cases?
Yes, I think if I'm not mistaken, most laboratories are able to do culture, but as we know, culture takes time. So tomorrow I'll be talking about alternate ways which are faster in identifying these multi-drug resistant bacteria because there's already a lot of commercially available kits, for example, or PCR kits that are able to detect, to give you a result in a quicker way than culture. So I'll talk about those detection methods tomorrow.
Okay po, thank you, ma'am. So we have another question from DOST ITDI from Miss Alma Cruz. Her question is drug resistance of an organism irreversible?
It depends because if they're plasmid borne, meaning if the bacteria acquired it from another bacteria through plasmid transfer, plasmids can be lost by the bacteria. So the way we want to maintain it in culture collection, if you want to maintain the phenotype, is you have to either lyophilize your bacteria or keep it in the presence of the antibiotic or the drug. But for those organisms which have acquired the resistance through genetic, like incorporating into the chromosome or the gene of the bacteria, then it becomes like part of the bacterium itself. So that sort of becomes permanent.
Okay, thank you, Doc Lu. So we have another question from PLM by Patricia Ann Marqueses. Her question is, hello, Doc. What are the diagnostics used when determining which species of bacteria has infected a person?
So just to give you a glimpse of what I'm going to talk about tomorrow. So we have, of course, the PCR, which can be used for detecting drug resistance in bacteria. And then we also have, of course, the whole genome sequencing. And then you have other ways of also determining through biochemical tests. So for example, if you have access to Vitec, they have a cassette which would allow you to identify whether your bacteria is resistant to a certain type of antibiotic.
Okay, so again, to our participants, this session has a part 2 tomorrow. So don't forget to tune in, same time and same Zoom link po ang gagamitin natin for the part 2 of Dr. Nadala's presentation tomorrow. And we will still be also live via Facebook at DOST ITDI Updates and via YouTube at DOST ITDI. So one last question before we end the first part of Dr. Nadala's session. The question is from Jimmy Bell John Donya of CMU. Her question is it possible to mutate some microorganism that combats these multi-drug resistant pathogens, like the antibiotic penicillin came from a microorganism?
Is it possible to mutate some microorganism that combat this? Maybe it would take time because as I have said, bacteria continually mutate, but the frequency is could vary from very high or from very low frequency to high frequency, but it's very hard to rely only on the mutation to combat this multi-drug resistant pathogen. So we need to develop more antibiotics or develop new antibiotics or develop new methods of developing an antimicrobial drug or antimicrobial substance, basically. So again, I will go over this tomorrow. I wanted to give a background on all these antibiotics and multi-drug resistance development so that we will be able to understand tomorrow how we could detect this multi-drug resistant strains, and also how do we handle them properly or better in the future when they continue to develop drug resistance.
Okay, thank you very much, Dr. Nadala, and to our participants who actively engaged in our Q&A session. So again, if you still have any questions, there is a part 2 of this webinar session on proper handling of multi-drug resistant bacteria. So that ends our session for day 1. Please be reminded again to attend tomorrow's continuation so that you can answer our evaluation form and secure a copy of your certificate and our speaker's presentation. But before everyone goes, can I please ask everyone to turn on your cameras for a brief photo opportunity with our DOST Balik scientist, Dr. Lourdes Nadala. So hello po sa ating 200 Zoom participants. I hope you have a camera so that you can be included in this rare opportunity. Okay na po ba? Naka-on na ba yung mga cameras? Can I check? Okay. Okay na po. On na. So I guess I will count so that our tech crew can screenshot every panel of this webinar session. Ready na po ba lahat? So can I get the bright smile of everyone? 1, 2, 3, say cheese.
Cheese.
Okay, first panel done. Second po. 1, 2, 3, cheese.
Cheese.
Okay, third panel. Ilan ba to? 8 pala tayo. So, third pa lang. Okay. Smile lang po tayo. 1, 2, 3. Say cheese. Okay, fourth panel. 1, 2, 3. Say cheese. Fifth panel na po. 1, 2, 3. Smile. Okay, sixth panel. Smile po. Seventh panel. And finally, the 8th panel. Okay, smile. Okay, thank you very much po for actively participating to our photo op. May picture na po kayo with our DOST Balik scientist. So again, thank you very much, Dr. Nadala, for your the first part of this presentation. Thank you very much for your in-depth presentation about multi-drug resistant bacteria. And see you again po for part 2. Again, thank you very much.
See you tomorrow too.
Yes po, thank you very much, Doc Nadala.
Mabuhay po kayo from BMUF.
Ma'am Lu, PSM 50th.
Oo nga, attend din ako.
BMUF po.
Sige.
Salamat.
Thank you, ma'am.
Salamat.
Thank you po. Goodbye, everyone.
Pasok, paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap liwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin. Sa pinagsamang lakas at galing, tagumpay ay mararating. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.